Brandon was born and raised in Philadelphia, Pennsylvania. His interest in horticulture started at a young age at the Philadelphia Flower Show. As a teenager, he developed a large collection of plants, mostly cactus and tropical plants. During his high school and college, he worked in ornamental greenhouse production. He earned his bachelor's degree in horticulture at Temple University, and after his graduation was promoted to greenhouse manager. Currently, Brandon is a master's student in plant breeding at North Carolina State University, where he works with Stevia. Please welcome Brandon Cooper.
Thank you, Betsy. It's a big pleasure to be here from NC State. Thanks to Iowa State for having me, and everybody who set this whole conference up. It's a pleasure to be here. So I from North Carolina State University, working on stevia breeding. So I'm going to go through this. I will mention that a lot of the work that I do is confidential. So I will focus more on yield components more than the sweetness profile that stevia is bred for, but I will be able to go through some of that. So here we go. So in 2014, we started off looking at stevia. My advisor, Dr. Todd Weiner, and I had no idea what stevia was other than it was a plant grown as an herb. You know, you go to a garden center, you have it, it's in with the herbs. It's just a little horticulture plant that you're going to grow in your garden. Plant's really sweet. It's a botanical name, Stevia rebaudiana. It's a native of Paraguay and Brazil in the Asteraceae family. It is a herbaceous perennial, and it's hardy to zone 9, so more for the southern climate. Being from Paraguay and Brazil, it is in high elevation, so it does get some hardiness to it, but it is kind of subtropical. The biggest component of stevia is that it can be used as a natural sweetener. And then again, there's limited research on this crop. So when we started off, we really didn't have a lot of information to base our work from. So we started off just looking. I arrived in the fall of 2014. We planted out some stevia plants. We got a first little look at it. The plant is very undeveloped and wild. High heterozygosity among the plants. A lot of diversity, just variations in leaf size and shape, and there's no true cultivars. So if it is a crop on the rise, especially in the south, but there's no variety that you would refer to plant if you wanted to grow it. So you're stuck with seeds, and as I'll show you later what seeds look like and the difficulties with that. So again, where do you start with a crop that's very new and needs a lot of work? Looking a little bit at the history of stevia, you can see where it was first identified where it's highlighted down here in 1937. The first PI was labeled of stevia, and then this graph over here shows that it was used by the indigenous people in Paraguay and Brazil roughly about 200 years ago, but it wasn't really documented until about 1899, and it was actually recorded as Eupatorium, which is a very similar Asteraceae. And then it was changed over to 1905 as Stevia rebaudiana, which is still known today. The key, 350 times sweeter than sucrose was identified in 1930, that this plant had a lot of potential in comparison to sucrose. And as we got closer, it was started to be used in China in 1980, and then in 2008, it was approved by the FDA in the United States. So a lot of the work done in stevia didn't start until about 2008 or after. However, China, Japan and India has been working on the crop for a little while now. Again, crop importance, glycosides. The steviol glycosides is what makes up the sugar, the sweetness in the plant. You also get bitterness with that as well. 300 times sweeter on average than sucrose. And the sweetness in the stevia is found among the whole plant. So you'll get sweetness in the roots, the leaves, the stems, and the flowers. So throughout the whole plant, but it is highest in the leaves. 0 calories, a big component of that. There's, it's quite healthy for you. They haven't found any negative effects on it. You know, it's being evaluated as a sweetener for candy and drinks, and you're starting to see a lot of products turn up with you know, sodas sweetened with stevia, because they can sweeten the sodas and things that have them essentially be 0 calories by a natural source, which is a huge demand in, you know, for health. So you see over here on the graph here that it is, you know, a natural. Stevia is natural. It has no carbs. It even has some dietary fiber. It has 0 glycemic index, so diabetics can eat whatever they want amounts of it. Some crop problems. Like I said, high plant variability and high phenotypic diversity. We started looking at different seeds, so we just gathered up whatever seed we could get from sources across the whole world and planted them out. And we quickly, you know, learned low germination rates, as some reports show, low sweetness in most plants, and there's a lot of bitterness. So you'll have a sweetness, but you also have this bitter aftertaste that kind of compares it to like Sweet and Low, there's some of these artificial sweeteners, which is not so desirable. But again, this is natural, so you know it has those benefits there. Poor branching, it tends to be very tall, upright, and brittle. The hardiness is kind of, you know, it could probably use some more winter hardiness, but not only that, it is from an area where it doesn't get so hot. So during the heat of the summer, plant flags a lot, and some plants flag more than others, so it has an effect on their growth. And then diseases, especially southern blight in the south, is a big problem. And as you'll see here in this photo, this is all from the same seed source, this plot right here. You have a plant right here, dwarf, small, you know, small leaves, just real compact, and over here you have one that's just dead, you know, a lot of just variation. I mean, you have, you know, ones that are real branchy, ones that are tall. So this is what you see. This is from the same seed source. So you bought a pack of seeds from wherever, and you plant them out, and this is what you see. And I see this with almost all the seed sources that we grow. So you know, that's, you know, for a mechanized system in the future, this becomes a challenge. So again, cultigen evaluation. We considered all the seed sources as cultigens, and we want to look at as many of them as we could. There's, you know, again, there's limited options for stevia germplasm. You're stuck with basically seeds, although you are starting to see some varieties that are patented in the past year or two that just came out. So there is some work being done. There's you know, again, a high phenotypic diversity among seed, and the big question is, you know, when you buy a pack of seeds from wherever company, where do they get their seed? Some companies will tell me they maintain their own population, but others won't tell you at all. So, you know, and just looking at the GRIN system here, there's no PI accession available for stevia. So, again, limited sources there. This is the variation that we see in stevia, just looking at the different seed sources that we find. I mean, just huge variations in leaf size, you know, the edging, you know, some have a serrated edge, some are more smooth, some are long and skinny, and, you know, and some of them have quite large leaves, up to, you know, 10 centimeters long, but on average, they're maybe about, you know, 5 centimeters, 6 centimeters long, and usually about 2 centimeters in width. So this one on the far corner here is pretty interesting in the size. You know, that's kind of, you know, that may be a component of, you know, what you want for yield in, you know, being able to produce the most stevia product possible. So we planted out stevia, all the different varieties, in a commercial planting, and stevia is planted in a commercial density of about 30,000 plants to an acre. So it's about at a 1 foot spacing. We planted, but we broke ours up into separate plots in that commercial spacing and planted them out. We had to do it all by hand because there's really no, you know, mechanized system, and we're planting all these different varieties. So, you know, we planted 30,000 plants by hand out in here, and then this is later in the season, the plants grow, and so as you can see, they get pretty tall. That's close to harvest. Sometimes they'll get almost about chest height in some cases, depending on your weather. Just looking, again, through some SAS data, looking at the variance, the important thing here is that, there was a huge location effect. The cultigen also had a huge effect, even though there is variation between varieties as well. And then, this is the interaction between location and cultigen. And this is for plant size, so we measured the plant height, width, leaf size, and leaf width. And then the bottom here is pretty much the same thing applies for the, the significant on location and cultigen and the interaction. This is looking at fresh weight. So after the plants grew out for the season, we mowed them down and we harvested, we got the fresh weight per plot. So then this is us cutting down the plots at the end of the season. We let them grow all season. Typically, stevia can be harvested 1, 2, or 3 times a year. When you cut it at the base, it does flush back. So, you can cut it twice, you can cut it 3 times, and you'll get, it depends what kind of harvest you're looking for. We just let it grow all season and got the fresh weight. And on a 30 plant plot weighed on average, about 30 pounds. But some, there was definitely some higher and some lower, and at the fresh weight. So about a 1 pound per plant. We planted each cultigen. We planted out in 4 replications at each location, and we had 2 locations, and there was 30 plants per plot, and then there was also a 6 plant plot where we looked at the sweetness compounds specifically for those varieties. Again, so, for the future, is this a clonal or a seed crop? There's a high variation of plants, again, for a mechanized cropping system, and seed is inconsistent in performance. This is the typical seed germination rate that you're looking at here. It's about, you're happy if you get 60%. And, you'll buy seeds stevia, and it's pretty low. I mean, you'll get 25% in some varieties or 50%. That's on average. And that to me seems like one of the lowest germ rates that's like acceptable for a crop. Imagine if you bought corn seed and you got 25% germination. Nobody, what are you going to do with that? So again, clonal propagation is likely to be the future of this crop. Here's the seed size right here, really small seed. Tough to work with. They're attached pappus, the Asteraceae family. It's hard to collect. The wind, you try to collect at the end of the season, it's windy, and the seeds are blowing away as you're collecting them, or they blow away before you collect them. So, so that's some of the issues. Looking at seed versus clonal, there's the seed plot right there. This is kind of early season. You'll see like the variation in size. Just, some plants are just small, some are big. And then clonal, these are just clone plots of some early selections that we made, and you can see how consistent they really are. So for a breeding method, we looked at hybrid field cages where we basically enclosed plants out in the middle of the field. We set up 10 pollination cages and 5 cages with bumblebees and 5 with blue bottleflies. We put 2 cultigens per cage, and we put 20 plants per cultigen in the cage, so 40 plants per cage. We crossed cultigens for agronomic traits, such as tall versus tall and large leaf versus tall. We also, we weren't able to do this yet because we didn't have the data, but in the future we'll include, the sweetest plants with the yield component as well, so that we can pull those 2 together. Usually they tend to be negative correlated, so you have a really sweet plant and you have really poor, yield component being that the plant's small and just poor performance. Just looking at, there's some examples, tall stem and large leaf. Compact, short plant, large leaf, and then you have some that just grow tall, kind of like corn. They just go up. They have minimal branching, but it's not always the case. They're highly variable. So these cages we set up, we did, this is the layout here, where we put there was 2 rows onto a bed, and we basically, like plot 901 right there, had variety in line A was 1 cultigen, and line B was another cultigen. So those 2 were together. The red line shows where we caged the whole block there. So those 2 beds were enclosed in the cage. But variety A is all the same, and variety B is all the same, so that we had the best kind of crossing going on in there. And then we selected the seeds. We harvested seeds the A times B, and then B the reciprocal, so with the other parent as the female. This is as the plant started to initiate flowering, we put the cage up. These are, we got these cages from Redwood Awning in California, and they're basically a fabric place. They made us these custom cages that fit our work, and then once they started to flower, we placed the bee box in there. So basically a beehive in a box, we put it in there. You see a lot of rain there. There's a lot of flood there. It was very rainy in North Carolina this summer, so constantly raining. So that's why we put them up on a cinder block. Stevia likes a lot of water too, so that's good. In Paraguay, it'll typically get 10 inches of rainfall per month. Here's a little better picture of the cages from the outside, and then again in the inside where they are actually starting to flower. So seed yield results, typically we'd get about 60,000 seeds per row bed, and about that whole cage, 40 plants, about 120,000 seeds. Again, these are small seeds, so you have like a gallon Ziploc bag filled with seeds, and that's like at least 120,000 seeds. It doesn't look like a lot, but it is. We collected the reciprocal crosses separate, and most seeds were fertile from what we found. Fertile seeds in stevia are generally black, and if they're brown, they're not so good. I had a lot of encounters with black widows this summer. There's one crawling out of a seed bag after bringing it back from our research station. When we were putting the cages up, there was black widows in the stevia plants. There was also nests in the corners of them, and then there must have been so many that they're even harvesting the seed. They came home with us, so that's kind of scary. But luckily, nobody got bit. So pollinators, we have bumblebees versus bottleflies. So I was curious to see, well, these flowers are so small, so is bumblebees effective? Are the flies more effective? So I kind of just estimated gathered up the seed totals, and on average, the flies had a little bit more seed production per plant. Again, not really statistically significant probably, but the flies, the good thing to note, though, the flies were easier to work with. See, the bee cage was cool. You just put the bee cage in there. They come out and do their thing, but bees are temperamental. They'll sting you, and plenty of people were stung by them. So that's a problem if you have people on your crew that are allergic to bees, and they really don't like to be in that cage. So they will get you if you try to go in there sometimes if they're not happy. But flies, you just put the larvae in there, in the cage, and they hatch in a day, and then the flies do their work, and they only live about 2 weeks, so you got to replace them, but the cost is lower. So you end up spending the same, but the flies don't sting you, and they're easier to work with. Although they are raised on fish, rotting fish, so they kind of smell bad. So we looked at the germ study to see were these seeds actually effective in being crossed, and yeah, they certainly were. We looked at, in our cages we had, cultigen 1 versus 2. We also did some where we put the same cultigen all in a cage to see what kind of, cross incompatibilities we had, because stevia is supposed to be self-incompatible. So we wanted to see, well, how close can we get to, selfing this plant? And that becomes a problem when you're trying to breed it, and that's why clonal may be the priority, because if you can't self the plant, you're always going to have this heterozygous population. We wanted to see the pollinator effect on seed viability, bumblebees versus bottleflies. It really didn't have an effect much, although, like I said, the flies were easier to work with. And then we have our open pollination set up where we just let the plants intercross freely, so it's essentially a wide polycross. There they are planted out in this beginning of the season. Again, this year we really didn't know much about the plants, so we just planted out all the varieties in a big block, and we got a lot of seeds. So this is the field later in the season. We collected seeds, so they're all half sibs, essentially, and but it's just a random polycross. In the future we'll look at more. We'll put actually plants out there that we really like by clonal selection or from its seed, and we'll have actual pollination blocks that have good stuff in it. We did greenhouse cages inside the greenhouse as well. We grew the plants out to flowering. I could fit 190 cages in my greenhouse. There's the small cages, just 2 cultigens to a cage, 2 per cultigen, and I paired them similar to the field cages. It allowed for a lot of crosses, and I just used the blue flies because I wasn't messing with bees in the greenhouse. Here we are, the plant's growing up about 2-3 feet tall, and then I put these tubes, these are like breathable, these are similar that they use in carrot breeding, so we kind of found that online and just used the same thing they do. Put the cage on when they started to flower, and then we secured the bag at the bottom. We set the pots on, and then we just kind of tucked the fly larvae in there underneath, close it off, and let them do their thing. And as you'll see, the fly is actively pollinating the plant. There's the little flowers there. And then looking at, for future studies, again, better selection. We'll have actual selections. This whole year we really evaluated all of our seed sources and got to see what we were looking at. And then we made a lot of selections from that this year. And next year we can actually pair those selections up and do our greenhouse crosses from plants that are really high in sugar or really high in yield. Again, they're separate selection criteria because of they tend to not be correlated. And we'll have efficient breeding strategies on stevia. Looking for this year, I'll have a whole season this summer to do that, to finish out my master's study. And we're going to evaluate the cultigens once again for another study to publish that. A lot of the work I do is pretty confidential, so I needed something to publish. So the cultigen evaluation was a little more open on the table. So and then we're going to use our clones. We're going to plant out our clones. We have some pretty cool, really high sugar content plants that we're going to plant out and evaluate them as cultivars. So and then we're going to look at we can continue to look at the germination pollinator effects. And just to wrap everything up, stevia is a very valuable crop. We're developing breeding strategies for new crops. It's kind of difficult to get started, but once you do, once you know what you're doing, it takes off, and it's great because stevia is such a new crop that there's a lot of work to be done in it. So we find ourselves always, getting sidetracked on things, and we really can't. We try to keep focused on what we're trying to attain. The overall goal was to make some germplasm available, release some cultivars. I am funded by PepsiCo, so that's the end goal, to produce a variety. With high yield and high sweetness. Yield is a tall plant, a big plant that also has high sugar. You can have a small plant with high sugar, but you can also have a tall plant with low sugar, and they might equal. At some point, there's a meeting point there. So again, you want want a lot of sugar in the plant, and you also want it to be yield. You want the fresh weight to be really high, and that sugar to be high as well. We'll continue looking at seed production methods and see if seeds are probably going to stick mainly with clonal stuff just because of all the difficulties we encounter with seed. But we want to see what exactly is the reason for the low germ rate as well. So with that, I'd like to thank Dr. Todd Wayner, my advisor, NC State Horticulture, and the research stations that we work with, my whole breeding lab with Dr. Todd Wayner, and the project funded by PepsiCo Global Research and Development. And with that, I'll take any questions.
All right, because we're a little bit short on time, we're just going to ask one question. We had a question from our online viewers on the webinar, and I do have to apologize to those listening on the webinar. We had a little bit of some technical difficulties, and I apologize for you guys not being able to see the slides as Brandon was talking. I will check with Brandon to see if we have permission to send his slides out afterwards, and if we do, we can get those to you, or we'll flip through them at lunch so you can see them. The question online is, it's come from Keith from OLTV. Do you think stress affects sweetness or picked up on any other environmental effects yet?
That's a great question. Yes, stress does have an effect on sweetness. We do note that the sugar content is the highest in the summertime, and I would be pretty sure that it would also be affected by stress. If the plant's stressed out, the sugars are probably lower, or maybe, we don't really know exactly yet, but they could be, I'm sure they are affected. Whether they're probably lower, but they could be also higher. So I can't really answer that, but I do know that the sugar is the highest at the peak of the summer, at the longest at the solstice.
Okay, so yeah, like I said, if you have questions in the audience here, please find Brandon during lunch or over break, and you can ask him those questions. But let's thank Brandon one more time.